And we're live.
Hello, hello, and welcome everyone to another webinar. We are so happy and excited to have you here with us today. I have an amazing panel. I'm so happy and excited to have our co-workers here on this beautiful webinar for today, where we're going to talk about the 3 steps to rapid soil regeneration, the soil food web approach made easy. So some of the names you already recognize, like Brian and Nick, and I think that Dr. Katerina is our first webinar with us, Dr. Katerina, so welcome, welcome, and welcome. As always, please type in the chat where you're joining us from today. I see that you already have people from Canada, Indiana, Massachusetts, UK, Portland, so welcome and welcome. Good day, good afternoon, good evening, where are you from today? So this webinar is part of the Earth Day awareness. As you guys know, next week is the Earth Day, April 22nd, and the world is just swapping in many amazing tasks and actions, and you guys are helping us making our Earth Day awareness. So thank you so much for being here. This is 4-part webinar series, and the series is called Regenerating Our Planet from the Ground Up. So as always, you guys are an imperative part of this journey. Let's just recap a little of the rules and guidelines we have in our webinars. So introductions will take about 10 minutes, and this presentation today is divided in 3 parts. First, we're going to have Dr. Katerina talking about how gathering the microorganisms. Then we're going to hear from Brian how to multiply the beneficial microorganisms. And Nick, we're going to tell us about how to apply the microorganisms. So as you see, the webinar today is really, really hands-on, and you're going to hear from our beautiful mentors. Then we're going to have a quick video about our current offer and followed up by the Q&A. We're going to be together for about 2 hours today. Nick, do you want to introduce the guidelines for us?
Yeah, absolutely. So look, guys, you're all going to be muted. And we would love to see loads of questions in the Q&A section. So please start putting your questions in for Q&A. We've seen some amazing questions in the last couple of webinars we've had. So please keep those questions coming to us. It's your great opportunity for you to ask all those things that you got puzzled in your mind about how to do things and things you're not sure about. So please put all your questions for the panelists in the Q&A. And as I say, we always like doing the chat as the presentations are going. It's really great to see what's happening. So yeah, brilliant. And also, Carla, because we're so full today, I think it's really important. I'm going to be paying close attention to everybody's time that they're speaking, just to allow all that time for questions, because we kind of ran out of time in the last couple of webinars. So I'll be watching the time for Katrina and for Brian as well today.
With the British clock, thank you so much, Nick. That's
absolutely
very important.
So without further ado, let's kind of introduce our panelists, shall we? Dr. Katrina Capri, please introduce yourself.
Thank you, and thank you everyone for being here. I'm very excited to take part into this webinar, which I believe it's gonna give us all and you all a quite great and broad overview of what it takes to start regenerating our soil. So I'm Caterina, I'm from Italy, as my names and my pronunciation is giving. I'm an agronomist. I've been working with vineyard cover cropping in the past years, and I've also been a mentor of the school in the past 2 to 3 years already now. And yeah, nice to meet you all.
Brilliant, Katrina. Thank you very much indeed. And Brian.
Yes, I'm Brian Dobbins-Beck. Again, very thankful and excited to be here to connect and share with all of you. I've been farming and consulting for about the past 11 years in conventional systems, organic systems with vegetables, fruits, animal husbandry as well across the US and Latin America. And I've one of the rather new additions to the Soil Food Web as one of the mentors, joined late last year. And again, excited to be here. Thank you very much.
Brilliant, Brian. Thank you very much indeed. It's great to have you both joining us on this amazing webinar we've got today. And also, we can't do a webinar without the amazing Dr. Carla Portugal. Carla, please tell us about yourself.
You were always kind, Nick. Thank you so much. I'm Carla Portugal from US, raised in Brazil. Portugal is just the family name, so it's the globalization everywhere. But the environmental science in my life just pulled me towards the soil realm and how we can and we must recover the soil, but we cannot lose track of biology. So my background always helped me connect all these bridges like Dr. Elaine does brilliant and amazingly. And I'm fortunate to be on her team, like we all are, and to help you guys as our amazing students to understand all the beauty and the benefits of learn how to manage a microbiome and recover the soil. I'm a science lead for the school and a mentor as well. And with our beautiful team, we are on the school for almost 6 years now, helping you guys help us change the planet. And now working in Oregon in Brazil, but like Brian, America, it's too big, so we can expand a lot of spaces. And what about you, Nick?
Thanks, Carla. Great words. Really appreciate you joining us again. So yeah, Nick Padwick. I'm a farmer from the UK, and I'm a soil food web consultant. Got my own business, Wild Soils, and we support, guide any farmer on that journey. Anyone who's really keen to explore these great opportunities. And I've been a soil food web mentor for the last year, I suppose, but thoroughly enjoying this amazing group of people. What such a great community to be involved in. So yeah, that's me. Okay, Carla, let's move on. We're getting tight for time.
Yeah. So like we said, this is a 4-part webinar series. We already have 2 amazing webinars. We highly, highly encourage you to go to our YouTube channel and rewatch them. The first one was with Didi per house. Didi is insane. There is a lot of beautiful techniques and tips and incredible things you can do in parties with your friends as well. So please go and watch. And the last webinar was with Nicole Masters, and unfortunately I was not there, but I watched it and worth every single second. So again, go back there and check. Today you're gonna hear from this amazing panel how we're gonna make the soil change for good. And next webinar is another house party. We are so excited to have this back, to be able to be there listening from you guys. You will be the star of the day. We're going to have multiple rooms. In each room, we're going to cover one different topic. You can jump in and join in each room and stay as many times as amount of time you want. But it's about to hear from you guys. How are you helping changing the system? How are you changing the soil around you? And it's all about you. So do not miss that.
No, you can't miss this next one. It's going to be a great day. Okay, I think we've covered all that off, and we're just about spot on time.
Yes. So, Katerina, it's up to you.
Hello again, and I'm very happy to start from the very beginning of the whole process, right? Because when we think about regenerating a soil, we have to start from the very beginning. Carla, if you could go to the next slide. Thanks. Because even though I know I should talk about identifying and gathering microorganisms, I really need to really quickly go through this, because we have to keep in mind that when we talk about soil regeneration, we need to take into account the soil food web, because there's no soil regeneration without regenerating the soil food web. It is indeed, as you might already know, right? Because the school is famous for that, Dr. Elaine is famous for that. And soil food web should be known from the very young age. But the soil food web is the network with all these micro and macro organisms that are interacting, right? All these pieces of the puzzle that are essential pieces that need to work to allow nutrient cycle, to allow a system to function, to be resistant and especially resilient. So whenever we approach the regeneration of a soil, of an ecosystem, we need to focus on the soil food web. And so that means that we need to try our best to enter in the system we want to regenerate the biology that is most likely lacking, or it could just not reach the numbers, the biomass that we need to have. And so we can do that, as Brian and Nick and Carla, with their experience, are going to share with us later today, by adding compost, teas, or extracts. But that compost, that tea, that extract we're adding needs to be full of organisms, and those organisms we want to have in the system. And so to do that, it brings us back to the main point. How do we gather microorganisms, right? It is at the same time pretty easy, but quite tricky as well. So that is something we want to work on, both from like a more theoretical point of view, but then also with Nick and Brian and Carla that are welcome to chime in whenever they have tips and ideas and comments. We're gonna try to dive into this topic a little bit more. But of course, gathering microorganisms around us that we want then to input in the system cannot be done without being able to identify those organisms. So if you want to go to the next slide, Carla, the idea here is I want to make sure that it's clear that the way we do approach identification, it's not very specific and detailed. And that is not an issue. It's actually quite functional, and it is something we want to do. We want to just make sure we are able to identify those organisms that that are like essential pieces of the puzzle we talked about before, of the network, of the soil food web. And we want to make sure we know what they are serving in the system. What do they do? What part of the nutrient cycle do they belong to? And so what do they what kind of what do they decompose, what they're feeding on, what they are preyed on? So those are the aspects we want to focus on. And we want to be able to identify on one side those organisms that we tend to call as beneficial microorganisms, such as the one we see in this slide. So those we want to have in our system, but also be able to identify those that we would not like to have in high amounts, mainly because they are indicators of conditions we want to avoid as much as possible, such as, for example, organisms that are indicating anaerobic conditions. And so here in this slide, we see the 4 key groups of beneficial soil microorganisms, such as bacteria, fungi, protozoa, and nematodes. They have different characteristics and functions. We're going to see a little bit of information, but of course, diving into all this would take a lot of time, but that is something that is very well discussed in the foundation courses and during the mentoring course. So that is something we could focus on. But basically, when we think about the 4 key groups of beneficial microorganisms, we start thinking about bacteria, which are the ones that tend not to lack in the system. But even though we usually look at bacteria as pretty negative because they're very high in numbers and biomass, very present in the system, they do have a very important role. They are decomposing organic matter, and they are extracting nutrients not only from organic matter, but also from sand, silt, and clay. And they are super important in terms of building structure in the soil because they are forming microaggregates by gluing them up, basically because they want to protect themselves, but is then very important role. They do work for our soil structure. And on the other side, we see fungi. They also decompose organic matter, but they do have a little bit different taste because on one side, bacteria are feeding on organic matter, but more simple material. And on the other side, fungi, they feed on more complex material. So you see, they do have taste that are a little bit different, and they work their way through improving our soil in different ways, but like on a parallel like direction. So when we think about then protozoa and nematodes, they are not directly decomposing organic matter or, you know, extracting nutrients for minerals, but what they do is they do feed on bacteria or fungi, and that is allowing us to then enter in the system food that is plant available. How does that work? Well, basically, when a protozoa such as flagellate or the amoeba we see in the photo, the beautiful tested amoeba that is in the bottom right side of the slide, that is feeding on bacteria, and all the nutrients that the bacteria is containing is somehow used from the amoeba itself, but the excess nutrient is then released in the system. So entering the famous poop loop, and of course, making it available for plants. And that is especially important because most of this takes place around the root system because of plants, like roots are like releasing exudates. So we do have this incredible activity that takes place around our roots. And if it doesn't, we want to make sure we are making the right decision to allow this interaction to thrive as much as possible. And then we don't want to forget about nematodes we see on the other, like in this left side, in the bottom left side of the slide. We do have so many different types of nematodes, and again, trying our best to make it as simple as possible. So we want to make sure we know what that kind of nematode is doing, what it is feeding on. So who is it preying and what kind of nutrients is going to release in the system. In this case, in this photo, we see a bacteria feeder nematode, and that means it's feeding on bacteria. And again, we said it for amoebas, but that is true for nematodes as well. So that it goes again with the poop loop and the nutrients being available for plants, which is pretty great. We can move on to the next slide, and we can dive into a little bit more specific information about those groups, as we also see some photos. One thing I haven't said yet is that I think it's quite clear from this video, and this is one of the reasons why I like this video, is because it's showing already how important it is to have diversity. So in this video, which is coming from, I think, a tea from a student of ours, we do see different shapes and behavior of bacteria. We see those rod-shaped bacteria moving around, some of them faster than other. We do see the more roundish one, so the cocci and so on. So that is quite important to keep in mind. I would say diversity needs to be one of the keywords to keep in mind in all the steps we're going to see today, not only in terms of biology and the microorganisms we want to have in the system, but then that is translated in the diversity of the material we're gonna look for and use for, and the food we're using, and so on. So diversity needs to be written on the stone, I would say. Okay, I don't know, you guys, if you want to chime in, please, if you have anything you want to add. Otherwise, we can keep on going. Okay. This is a very beautiful fungal IFA here. I would say it's the perfect IFA. When we think about fungi, we do think about this very nice dark brown, homogeneous diameter with nice septa, which are the cross walls. And this also has the bumps that are called clump connection, which is also very, very good indicator of a good guy fungi, as we like to call them, right? So those are, as we said before, decomposing organic matter and are consumed by predators such as nematodes. We talked about nematodes before, but the photo we saw in the slide before was about a bacteria feeder nematode. And we do have different nematodes feeding on different microorganisms, and one of them is a fungal feeder. So they're feeding on fungi, and that means that is making the nutrients that is in the fungi available for the system. So that is very important. And as we said before, not only fungi are decomposing organic matter, but they are also actively improving the structure of the soil. differently from bacteria, as we saw before, bacteria, they're improving the soil structure by gluing microaggregates together, right? But on the other side, fungi, they do have this nice filaments that are really making a work towards like creation of macro aggregates. So there's a different level of aggregation happening. And of course, they do retain nutrients, as we saw before as well. We can move on to the next slide. Here we finally see a lovely, again, amoeba. I think we're quite fan of amoebas. At least tested amoebas for me are super pretty, super fascinating. And I think they scream diversity, as we said before, because it's quite easy to see them with those lovely shapes, crazy test, which are, you know, the container, we can say, where they try to protect themselves. And in this case, I really like this video because it's showing the amoeba itself, because it's like showing the pseudopods, which is coming outside, allowing it to move around, to grab the food and so on. As we can imagine, we do see bacteria in this video as well, protozoa, and so tested amoeba as well. They feed on bacteria. And then, of course, then they release the nutrients because bacteria have denser nutrients concentration. So the amoeba doesn't need all of it. And so it's releasing the excess, which is, as we said before, like made available for plants. And we didn't talk about psyllids. You can see a note here on the side. Just because we talked about protozoa such as flagellate and amoebas, which are the ones we tend to want to find or have in our system. As we said before, we may and we need to be able to identify those organisms, microorganisms that we don't want to have in very high numbers, such as psyllids. Not because they're all bad, and we don't want to make this distinction between good guys and bad guys, because as we said before, everyone has its role in the system, but mainly because we want to keep in mind that some of those microorganisms are indicators of conditions we want to avoid, such as anaerobic conditions. And in that case, psyllid, they do thrive in lower oxygen conditions, which is something we want to avoid in our systems. Okay, I think we can move on. Carla, thank you. Okay, this is another lovely video. I like it, though it gives us the perception nematodes are huge, which sometimes is true, but sometimes is not that evident. But this is a very nice video showing us, in this case, a bacteria feeder. Bacteria feeder are feeding, as the name says, and as we said before, on bacteria. But we may have different types of nematode. We talked about the fungal feeders. But we also have predators nematodes, which are feeding on other nematodes, either beneficial nematodes, such as bacteria feeder or fungal feeder, but we may have also predatory nematodes that are feeding on root feeders, which are nematodes we tend to be afraid of finding in our soil because they are attacking our roots and damaging the systems. So it's very important to be able to identify nematodes, to be then confident enough to say, I do have a nice nematode population in my compost and I want to enter it in the soil. So yeah, we can move on, I think. Thank you. Okay, this is a video of a fungal feeder, so fungal feeding nematode, and we can clearly see that thanks to where Carla is pointing at, we do see the spear. It's a very quite long and thick spear, which is used to punctuate the fungal strand, and you can clearly see that. That gives me the chance to say that the identification we aim for is based on morphological characteristics, right? So based on the mouth part and the inside part of the body of the nematode, we are able to understand what their behaviors are in terms of what they're feeding on. And so in this case, the spear, it's an indicator of a specific way of feeding on. While on the other side, the previous video showed us a nematode with a mouth that would not allow feeding on fungi or anything else than bacteria, because it did have a cylindrical mouth and so on. So that is also very fascinating. We can move on. And this is just a bunch of other organisms, microorganisms we might find. We have a naked amoeba on the top left side. We have a ciliate in the bottom. There's a lot of diversity. We do have flagellates. We do have a nematode egg and so on. A tardigrade, I would say. So that's of course very fun to see, quite exciting the first time you get the chance to meet them, and incredibly interesting understanding what they are indicating and telling you about the situation. Again, we do take a sort of photo of what is going on in the system when we do an assessment. So it is very important to get as familiar as possible with interpreting the information we may get from finding or not finding organisms. Okay, do you guys have anything to add?
I would like just to complement one very important, one of the many very important points you touched, is that we are worrying about the ecological role, not the species per se. So because we want to make sure that nutrient cycling is running as smooth as possible. So this is why we don't care much about learning about specific species unless you're doing a work with them, but concentrate really on how they are supporting the food web. So I think that's a very good point, Katerina. Thank you.
Okay. Well, we now have a little bit of an idea of how to identify them. And then, of course, once you know how they look like and what do they do, we start thinking about how can we gather them? Like, is it easy? Where do we find them? And so, as the title here says, where do microorganisms hang out? Well, it's gonna be maybe obvious for many of them, but maybe not. But well, we do have microorganisms all over the places, like around us, and not only, like inside and outside our body as well. And so just keep that in mind if that is the first time you think about this when you have a walk later today or tomorrow. Look around yourself and just try to imagine how much biology you may have around you in terms of microbiology. And so that is translating in, okay, so how can I get that microorganisms into my compost and then into the soil? Well, that is how you have then to start thinking about gathering starting material for making the compost. So microorganisms are, as we said, everywhere, also in the air, like in the wind, we have spores, we have like microorganisms. So inoculum potential is everywhere. And so again, when we approach like looking around for gathering microorganisms, we need to aim for diversity. We can move on to the next slide.
And Katerina, just quickly, sorry, hate to say this, you've got 5 minutes.
Ooh, okay.
5 minutes.
I'm gonna
Gotta
speed up.
squeeze a
I
lot
think
in 5
everything.
minutes.
Okay, so I'm not too worried, just because I would say all the next slides are things you're gonna touch on. So I think it was very important to make sure we had an idea of microorganisms and how they look like, and that the diversity is super important. So I'm gonna briefly touch on this because, of course, while we're gonna talk about composting, it's gonna be well discussed. But as you can see here, when we think about microorganism gathering, as we said, we need to think about gathering feedstock material to start a compost. And feedstock material needs to be as diverse as possible, not only because they function differently in terms of food and how they can work on that in that terms, but and also how they're gonna behave in the pile in terms of like texture and structure they can allow, but also in terms of what kind of inoculum potential they are bringing to the system. And as Nick and Brian are going to explain, we do have materials distinguished in high nitrogen, green, and brown material based on their CN ratios. So their carbon and nitrogen atoms they contain. As you can see here, green says, in my case, it was my prunings, like high end, it was like horse manure. And in the bottom, you can see wood shredding, so brown, because it's very high in carbon content. We can move on. I don't want Nick to be telling me I have to speed even more. But yes, one thing I really want to pass on you is that, again, diversity. But diversity is very important because as humans and as my cat, as you can see in this photo, also microorganisms have taste, like this different preferences for food, right? So we are not necessarily finding all the groups of microorganisms in the same feedstock material. And in order to have as diverse biology as possible in our compost and ideally in our system at the end, we need to start with a diverse food typology, right? And so that it does bring different inoculum, but it's also going to be a different food for different microorganisms needs. Okay, we can move on. That was a more serious couple of photos, but again, the same ideas. So we can move on to the next slide. And just so that we have a little bit of a better understanding, even again, it's going to be discussed later on. We talked about diverse feedstock because they are diverse food, and so they are diverse, like there's the diverse preferences from like different microorganisms. And as you can see here, for example, high nitrogen foods such as manure, beer mash, legumes, plants, alfalfa, and so on, they are very typical bacteria food. As we said before, you may remember we talked about more simple food, and that is the case. And that is similar when we think about green foods, right? Grasses, veggie scraps, and so on. Again, it's like very close to have a narrow CN ratio. That means easier to decompose. And so bacteria are the ones that tend to prefer this kind of food. And then we have the woody brown material. So those that have very high carbon content, and they are quite complicated to decompose and to feed on. And so then we need fungi to do the work in this case. And we think about wood chips, coconut husk, dry leaves, but also paper and cardboard. We, though, just a point here, have to keep in mind that, yes, we may have different foods, but not all the foods we can bring to the system are bringing inoculum potential. So we have also to think about it. And I'm sure Brian and Nick are going to discuss this later. So I'm not sure if I have one last slide. Okay, just so that we keep in mind that, yes, as we said before, gathering microorganisms, as you saw, it's pretty easy. It's everywhere, right? But it's not that easy sometimes. It can be tricky. It depends on where we are. Because we may encounter feedstock material that can be easy to access for us, but they may have some issues in terms of like effectiveness of the biology that they contain and how they could work as food. We may find like feedstock materials such as, you know, as you see here, pine needles and so on, that may contain antimicrobial like compounds. And so they may, instead of like allowing our microbial population to thrive, they can work the other way around and slow down their like metabolisms and activity. We also want to avoid areas that have been treated with pesticides and so on, because they do affect the microbiology, of course. And we also want, for that same reason, avoid manures of animals that have been treated in ways that may affect the microbiology present. That is the last thing, I swear, because I've been talking about diversity, but you know, the importance is to also keep in mind the context, because the diversity is important for that different reasons. Because if we enter in a system, a diverse population is giving to that system more resilience, right? Because each of the organisms we have or we may have in our system have specific needs in terms of humidity, in terms of temperature, in terms of food availability, and so on. And so in changing conditions, such as the ones, especially it's the ones we're facing right now, we need to have diversity. And so diversity, though, it's like sort of like auto-selected in the context. So like sourcing from around you is speeding up this process because you're already selecting microorganisms that are able to thrive and exist in the context you want to have them to exist and thrive. Okay.
Katrina, that is fantastic. And you were absolutely on your 5 minutes. So
that's good. It's
well
also
done.
pretty rare for an Italian. So I'm pretty proud of myself.
Well, that's great. We like it. We like it a lot. Thank you. We embrace that. We embrace it. So are we going to have some questions, Carla, or are we going to move straight on with Brian?
I think that do you guys have any compliment? I already put my 2 cents on Katerina presentation, so
No, I thought it was great. It gave everyone a great overview of what's happening and what you
yeah.
can do. And hopefully by the end of the webinar, once Brian has done his presentation and myself, people have more of an understanding, and hopefully
Yeah.
more questions will come in the Q&A from that. So without any further ado, Brian, over to you, please.
Thank you, Katerina. I guess the funny thing I would mention is that everyone can tell that's a lot of information to digest, and you're not going to remember all those details today. But I think the main point is the science that's behind this method. And a lot of research has gone into it. And in my experience, I haven't done that research, but I have used it practically, and it does work. And that's the thing that changes this program from every farmer has like a rule of thumb or like a heuristic. They'll tell you like what works, what doesn't work. But this program really ties everything together for me. It's almost like a unified field theory of agriculture. So when someone tells you something works, you can now understand why it works in their situation, and might understand also why it doesn't work in a different situation. So anyways, thank you, Katerina. And yeah, a lot of research and details and a lot of things to digest. So we talked about identifying these microorganisms. We talked about gathering them, and now I'm going to talk about multiplying them. This is just a quick overview, and I'll go into more details on each of these. But we'll talk about the soil food web approach, how that differs from other methods you might have seen. We'll talk about what biocomplete compost is. There's lots of different composting methods in the world, and biocomplete is something special that goes into dealing with those microorganisms that we just talked about. What the objectives of this compost are, it's not just nutrients, it's about the biology. We'll talk about some other key considerations. All of that will tie into the compost recipe and what we're considering as feedstock materials. We'll talk about managing that pile. One way we manage that pile is by turning it. And then at the end, we'll go over a quick equipment list that would be useful to get started with. Good. So the soil food web approach is both holistic and scientific. Holistically thinking, on Earth the Earth's been evolving, life's been evolving on Earth for billions of years. If you have a system created by humans, for example, an agricultural system, it's almost by design and necessarily not a natural system. So it's out of balance at the moment. And a lot of the natural cycles, whether that's the water cycle, nutrient cycling, such as carbon cycling or nitrogen cycling, those cycles are broken. The easiest way for us to address that and bring this into balance and make a farmer's life much more easy and enjoyable is to get these cycles working again, and we do that through biology. So that's how I think about it holistically. Scientifically, we're considering all of that biology. You're looking at biology, and you're using a microscope possibly on a daily basis to monitor your amendments and to monitor the system that you're managing. Just a quick note that this webinar series is going to provide a high-level overview. We're not, even though it seems like we're going into a ton of details, there's even more details in the coursework and even more hands-on experience in the CTP course. So again, this will be somewhat of a high level, even though it seems extremely detailed. Good. So there are some important factors into making compost. One, we need a complete set of the soil food web organisms, and we have those defined quantitatively in our manuals and our coursework. As Katerina mentioned, we're looking at bacteria, we're looking at fungi, we're looking at nematodes, we're looking at protozoa. We're trying to maintain aerobic conditions. The roots of plants, which is what we're striving to grow, are obligate aerobe. So the plant roots, if they're doing well, they're in an aerobic environment, and the organisms we want around those roots are also aerobic. We're just like all life on Earth, there needs to be some water, so we're maintaining adequate and consistent moisture levels. And considering that, not only And then when I discuss these, this is not just the compost, this is your agricultural system that you're also monitoring and considering. Whether you're talking about compaction on a roadway or compaction around plants, you're constantly thinking how these microbes are surviving. And then in the composting part, temperature is a key factor. We'll go into more detail about that, but that's basically about keeping the organisms that you want and then removing the organisms that you don't want, including weed seeds. So what are the benefits of biocomplete compost? One, it makes nutrients plant available. As Katerina mentioned, you have your bacteria and your fungi. And I guess before I mention that, most of the soils, the land around the world, the dirt, the earth, it has the nutrients to support plant life. The problem is that those nutrients are locked up in your sand, silts, and clays. They're not accessible to plants at the moment. Again, the earth's been evolving for billions of years. Life has a way of dealing with this. So when you have the biology in that system, you have your bacteria and your fungi. They release enzymes. They digest the food outside of their body. So they release, if I'm incorrect, somebody with a PhD will stop me. The bacteria and the fungi release enzymes to digest their food outside of themselves. That breaks down these sand, silts, and clays, and then they're able to absorb those nutrients. So the second point here, you're able to retain your nutrients better. Your nutrients are now in a biological form. They're moving around in the form of bacteria, fungi. The protozoa and nematodes are eating those and excreting those in the plant's root zone, giving the plants food. So all these nutrients are being broken down from the sand, silts, and clays, being put into a biological form, and then right where the plants need them. The plants are producing exudates that invite the biology that they want in the root system there. So it really takes care of itself. You're able to suppress pests and diseases. This goes to what Katerina was mentioning with diversity. When I think of a conventional agricultural system, if you're growing tomatoes, for example, or you've seen a field with tomatoes in California, perhaps, the goal of that farmer is to have one species of life in that entire field. He just wants tomatoes. He doesn't want another plant growing. He doesn't want any insects in there. They don't want anything else. Fortunately, the earth doesn't allow that to happen. So the earth has some bacteria in that soil, and the earth's pushing up some weeds. The earth's striving for diversity, but at the same time, the farmer's continuing to just grow those tomatoes. When you have a diversity of biology in your system, you're not as susceptible, you're not as vulnerable to diseases, you're resilient, as Katerina mentioned. So when we're putting out amendments with compost or teas, you're literally talking about tens of thousands of species, not the quantity of biology, that's in the trillions or more, tens of thousands of species of biology of life in that field. And then that's not even including if you have a ground cover going. But when you have a system like that, that's in balance, there's natural cycles happening, you can introduce on purposefully, a pathogen, and it's not going to be able to get a foothold. It can't take over. Whereas if you have just tomatoes growing and there's no other life in that thing, it's very vulnerable to an outbreak, and that's when you have to bring in even more destructive chemicals. In terms of building soil structure, Katerina mentioned that the microaggregates are formed by the bacteria and the fungi, and those lead into macroaggregates, and those aggregates form soil structure, which allows the penetration of air, water, plant roots. This system with this biology is also able to suppress weeds. When you have a bacterially dominated system in a conventional sense, in a conventional agricultural system, your nitrogen cycle is mostly putting out nitrate. Your low-level plants love nitrate. What we consider weeds love nitrate. So you're basically selecting and voting for weeds to come about. When you put out, again, these tens of thousands of species of biology and they're doing their thing, your nitrogen cycle converts to ammonium. Not only is ammonium less mobile in the soil, but it's what your crop plants want. So you get a lot of benefits from this biology in agricultural systems. At the same time, you're able to store carbon. That seems obvious. Your plants are thriving. They're essentially solar-powered carbon pumps. The life in the soil is carbon-based. You're able to sequester a lot more carbon. And then a variety of these this biology is able to at the same time decompose toxins. Good. If there's any other comments from anyone else, but if not. So what are our objectives? Our objectives are to, based on all the benefits we've been mentioning, we want to multiply or grow the beneficial microorganisms, and we want to eliminate or reduce the detrimental organisms. What's the science say? The beneficial organisms like aerobic conditions, the detrimental microorganisms like anaerobic conditions. And to me, the even simpler idea is knowing that plant roots are aerobic and we're growing plants and they feed from the roots. So we want aerobic microorganisms. Good. Next slide, please. So this is so again, we're attempting to multiply aerobic microorganisms, and we do that through these variables. And we'll go into more detail on each of these variables, so I'll just kind of briefly list them here. But temperature, we're considering, and then obviously the volume of the pile, the amount of nitrogen, the temperature you're trying to reach can change based on seasonality. So we consider that. We consider the moisture. Again, all living things need water. We're considering oxygen levels. Again, we want aerobic. We're balancing the biology for the specific crop that we're trying to grow. The fungal to bacteria ratio ties into so many things. One, which I mentioned, was the nitrogen cycle. So are we getting nitrate? Are we getting ammonium? And then what kind of crops are we trying to grow? If you're growing an orchard with trees, you're aiming for a higher fungal to bacteria ratio than you are if you're growing tomatoes or oats. And we can talk a little bit about that as well. All of this, again, ties into the right recipe. The recipe ties into each one of these. So the recipe is going to dictate your temperature, your moisture, your oxygen levels. For example, your woody materials are going to provide structure that allows air to enter that compost pile. And then not least is timing and turning, which tie back to temperature. So we're managing our temperature, and we have thresholds which we use to let us know when we need to turn the pile, and we'll go into those details here in the next slides. Good, so the first of this is temperature. So how are we monitoring temperature? We have typically a thermometer that we use to check the pile from different angles, from the top, from the sides, and we're maintaining that temperature. And we have a list of thresholds that, again, is based on research that's been done by Dr. Elaine and others. So the sweet spot here is between 55 degrees Celsius and 65, 75 degrees Celsius. I'm not sure what my audience is, mostly from the States or Europe, but I'll stick with Celsius for right now. So when the temperature is too low, the disease-causing organisms are not killed or forced to go dormant. So you end up keeping too many of those around. And the sweet spot is where you can kind of get rid of those and have your aerobic organisms thrive. When it gets too hot, you're going to one of your other variables and considering the oxygen levels. And someone else here might be able to speak to, I mean, if it was too hot, especially for too long, it can be detrimental to your beneficial organisms as well, I assume. So we're shooting for this sweet spot, and we're monitoring it daily. Good. Moisture levels. So again, considering water. The sweet spot here is around 50% moisture. How are we monitoring this? You can use some fancy, maybe a little bit expensive tool. What we tend to use is just our hand in a squeeze test, get ourselves calibrated for how many drops should come out of each squeeze to know what our moisture level is. And once you're monitoring this pile with a microscope, with your eyes, with your nose, with your hands. You start to get a feel for what it needs, and it's kind of an art and a science, and that's kind of the enjoyment of it when you get doing it. So 50% moisture is what we're shooting for. Less than that, microbial activity and growth are inhibited. And then when the moisture is too high, there's not enough room for air to enter, and again, we're considering that to be an anaerobic system. And then if it's anaerobic, we're helping the anaerobic guys thrive. So we're trying to keep it aerobic, good temperature, good moisture level. Considering in all of that we're trying to get these aerobic organisms to thrive. Good. Next slide. Oxygen. So the number that we often mentioned is 6 parts per million. That's kind of the threshold. Below 6 parts per million, we start to have anaerobic conditions, and we favor those microorganisms. Above 6 parts per million, we start to see our aerobic organisms. How are we monitoring this? We don't have there are oxygen meters and different things we can use for teas and extracts, which we do use. But at the same time, it's just as easy to get your microscope, look at what's going on in the pile, see what organisms are you looking at. You'll certainly know when you start seeing more ciliates or rotifers or worse things that your oxygen levels are dropping. So we mostly monitor this through the microscope. Good. Next slide. Considering fungal to bacteria ratio, I talked about this a little bit. In most agricultural system with most crops, you're looking for a balanced 1-to-1 fungal to bacteria ratio. This is good for mid-successional crops like vegetables, grains, perennial grasses. When you get to a lower fungal to bacteria ratio, you're somewhat selecting for early successional plants, some which you might consider weeds. And then when you get into, for example, an orchard or woody plants, you're looking for a fungal dominated compost or amendment to use, and that would be in the range of 5-to-1 or higher. Next slide. So the compost recipe, as it says here, it all starts with Dr. Elaine's compost recipe. If you get this wrong, your chances of success are very low. And even when we go through this in the CTP program and we're close to doing it right, if you're not paying attention to all those variables and not listening to the pile, listening with, again, your nose or your eyes or the microscope or your thermometer, it goes wrong. There is a recipe, there is a science behind it. So typically, depending on your climate, you're deciding what your ingredients are. If you need more heat, you're selecting for more high nitrogen materials in your feedstock. Our high nitrogen is typically about 10 to 20%. The greens around 20 to 30%, and then your woody brown material about 60%. Your high nitrogen is going to kickstart this pile and get the heat going. Your green's going to maintain that heat through the cycling. We're going to go through at least 3 cycles. And then your the wood brown material is going to provide carbon. It's going to provide structure to the pile so that air can enter. It's also going to inoculate with a lot of good fungal species that will thrive once that pile is maturing. Good. Next slide. So when do we turn the compost pile? We have these thresholds. We're considering temperature and moisture, and we're considering the oxygen levels when we make these decisions. So the first thing is the temperature, and we want to make it hot enough, long enough, so that we're killing off the detrimental organisms and the weed seeds. So each part of the pile, and we'll show that in the next slide how this cycles, but we want every little piece of that pile to go through a heating cycle. We want it to cook. So above 55 degrees Celsius, if it's at 55 degrees Celsius, we'll do 3 days of waiting and then we'll turn. If it's at 65 degrees Celsius or a little bit higher, we'll wait 2 days, 48 hours. And then at 71 degrees Celsius, we can turn it within 24 hours. And that's kind of the procedure we use. We're checking temperature daily. We're also monitoring the moisture daily. And after these cooking processes happen, we turn the pile to, again, reintroduce oxygen and then to get another aspect of that pile into the center of it so they can cook. And we'll show that here on the next slide. Good. So this is looking at this diagram. This is how we kind of make sure we're going through a heating center, cooking the center of the pile so that you have this spherical shape inside your pile that's being heated up. And that's what you're going to reach into with your thermometer to check the temperatures. So we have this hot center. We let it cook for a certain time depending on the threshold. Once that's done, how we rotate it is we remove the top portion of this pile. We set it aside, perhaps on a tarp. We take that hot center, we set it, now becomes the bottom of the pile, as you can see on the second pallet here. We then take the top that we removed, we place that in the center, that's going to cook this time. And then we take the bottom and we set that on top. And then you repeat that process one more time, and then you've now cooked all aspects of the pile. And after that final heating cycle, we'll at least turn that pile one more time to reintroduce oxygen. And if it does not continue to heat up, we will let it mature. If it continues to heat up, we'll continue to turn it, basically to make sure that we're not losing oxygen and multiplying our detrimental organisms. Next slide. So here's everything we're talking about right now is composting on a small scale, and we'll go into large scale composting after this. I meant to mention that earlier, but in terms of small scale composting, which we're talking about now, the tools that will use or need want to be a wooden pallet, something to get this pile off the ground, not to have it sitting. It's nice to have it wick moisture, not be sitting in a pile of water. A sturdy steel wire cage to kind of keep its form, a suitable water source. We're always considering the water that we're using for the pile. You're constantly thinking about these microbes and what's happening to them. So if you're putting chlorinated water on this pile, that's a problem. So everything you're doing, you're thinking critically about these microbes, how they're your pets. You're trying to keep them alive. You're trying to get them to multiply. 5-gallon buckets for measuring and also perhaps for soaking materials in, a sturdy tarp, a pitchfork, hose and spray nozzle, and lots of energy and good health. I think that might be the last slide. If there's any comments from anyone else before we get into large-scale compost production.
No, that's great, Brian. Not from me. Anything from you, Carla?
We have a nice tip as well, right, Nick? Name your pile.
Absolutely. People like to name their piles. Absolutely.
Yeah.
Absolutely. But
If you
guys,
are one of our students, just log in on the FC forum and watch the FC webinar we had early this week, and you will understand the joke.
absolutely. And as we move on to large scale composting, just to remind you all that on the 29th of April, I'm afraid you'll have to put up with Brian and I again, because we're gonna present a bit more, spend a bit more time on large scale composting, which is, I think, something that you and I are going to do a bit of a double act on now, Brian. Just a very quick one. We've got 4 minutes. 4 minutes to do this.
I didn't get my 5-minute warning, so I'm taking 6.
Yeah, well, I'll be in control of that, Brian.
Okay, I can go over this high level. So what's nice about this method, and it's definitely, I mean, having taken the CTP course myself, it's changed the way I farm 100%. And it scales up. So doing this with windrows is a lot of fun. There's very slight changes you make, but not much. You're considering all the factors that we just talked about. One of the contexts that I've used this method is on a organic orchard in California. It was a 280-acre perennial fruit farm. We grew organic fruits and nuts, very diverse, lots of different fruits. And I'll go into maybe some of those here in another slide. And the compost in this situation was for on-farm use only. So it's if you go to meet with a farmer, it's good to get a feel for what their context is. What are they using the compost for? Do they sell it? I wasn't selling it in this case, but it was for a USDA certified organic farm, so that changes some things. Do they purchase input materials? I did not purchase any input materials, and I didn't purposely grow any materials to feed these composts. And oftentimes when I needed really good compost, whether that was for new plantings or weak orchards, I would supplement that compost with worm composting that we did elsewhere. Kind of the why behind some of this. So why did we start? One, usually in a community and a farm has a lot of organic residue, which sometimes can be wasted. We were able to use this to cultivate and propagate microbes across the orchard for the reasons I've already discussed, getting that diversity back into the system so that it's more resilient. There's also marketing benefits that are kind of add-on to this, where there's now regenerative certified, which you can move your products into a higher price point. You also have access to other grants. And it's also just nice to know people often buy things from people that are sequestering carbon and taking care of the planet. And knowing that this biology is thriving, it definitely helps with marketing. Good. Next slide. So what were the ingredients that we used? discarded fruit from storage. So this particular farm, we harvested fruit extremely ripe. One of the benefits of that is that the fruit tastes amazing and better than anywhere else you get it. But at the same time, the downside of that is you throw away a lot of fruit. It goes bad in storage if you don't sell it right away. So we had literal tons and tons and tons of fruit that we could put in this compost pile as green materials. We had tree trimmings from our own orchards and from other landscaping businesses and orchards around that we would use. There were a lot of stables around, so we used local horse manure bedding. Coffee grounds from a coffee distributor. After those coffee grounds were spent, those work well as a green material. And then as needed, high quality compost from other piles, kind of like when you do kombucha or you're making fermented bread. What do you call it? I'm at a loss for words. Sourdough bread. Good. So you're using usually like a mother pile or mother system that has these organisms, and you're inoculating into your pile. So that's always important depending on the biology you're shooting for. And we did about 60 of these windrows a year. Each of those windrows are about 75 tons each, about 100 cubic yards. And that allowed us to put about 15, I'll go more into that, put about 15 tons per acre per year. Good. I'll let Nick take over. Thank you, everyone.
Thanks, Brian. That's great. If we can move on to the next one then, please, Carla. You've left me with like 30 seconds, Brian. Thank you. So look, brilliant principles. And listen, in the UK, I kind of follow the same. So we're using wood chips and then herbal lays. So we're using clovers within the lays, legumes are going on there, and then grasses and herbage for our green. And then we balance that out with our wood chips. And you can see the guy there making our windrows before we mix it up. And we also practice some of these recipes in my round towers, which you can see we've got different round towers there. Okay, Carla. When we're thinking about how we generate all our feedstock material, I'm actually incorporating that into my growing crop. So I'll plant in some herbage lays, and then I'll take my wheat crop. So the picture on the left shows the wheat crop standing, and underneath is the clover you can see and some other herbage. And then as soon as we've harvested that crop, we let it grow for a couple of months, and this is in the autumn now. And then we'll cut that, and I've got some great brown material, I've got some great green material, and I've also got some high N. I sometimes, because of the autumn, I sometimes substitute that with a bit more high nitrogen clovers just to make sure I get that heating process. Okay, Carla, thank you. And you can see as we start to mix the compost, the top photograph, we're going at a really steady pace. So we're probably going through it at about 0.1 of a kilometer an hour. So make sure if you're thinking about investing in this sort of equipment, make sure your tractor can go as slow as 0.1 of a kilometer an hour. Very important. And also from moisture. Moisture is also really, really important. We don't need as much, I find, in a big windrow because of the biomass as I would if I was doing a round tower. So that 50% is probably 40%. But I do it in the same way. I'll squeeze it, open my hand, and if it breaks open, it's about 40% moisture. So I'll use the totes I've got in the back of the composter, and I apply that water. And it could be as much as 30 to 40 litres a metre, depends on how dry my feedstock material is. Okay, Carla. Thank you. And there we are. We've got a nice windrow that's been completed. We've actually put 2 windrows into one. So I always make enough to do 2 windrows about 3 meters apart. And as they start composting, they reduce in size. And then I push one pile onto the other pile. So I've got more biomass. I think that's really important because it helps to retain moisture throughout the drier seasons or even the winter when I need it just to stabilize in the winter, ready before I start using it. Okay, Carla, thank you. And here's a great picture of our applications. We've applied some compost, and then kind of 6-8 months later, the picture on the right is that soil has slightly changed color, and you can see those fungal hyphae running through. Thank you, Carla, running through that soil profile. And Carla was with me in November, and we dug some of this soil up so she could see it. So, you know, that's what a solid amount of compost would look like. And we would spread anywhere between 2 and 5 tons, dependent on the biology you've got in your compost. And that's really important as you're on that journey to start keep on checking the amount of biology you've got in your compost. Okay, Carla, thank you. And then just to wrap up this, the large scale bit. So this was something we did last year. So April 2023. This was a typical field of ours, very high bacteria, very low fungi, and you can see there that my fungal to bacteria ratio was really low, and you know I'd got some no, on the actual slides, Carla, sorry, not on the S map, sorry. We got really low protozoa, really low fungal to bacteria ratio, 0.01. And then we start introducing 3 applications of extracts, and this is the S map at the end of it that shows where we are. So we've gone, you can see our bacteria numbers have dropped. They've dropped because the protozoa numbers have gone up. I haven't put the bacterial feeding nematodes in there as well, and our fungal hyphae numbers have gone up. They've moved up the scale. Get to that, getting closer to that 1-to-1. That's where I'm aiming for. I'm not there yet. I'm at 0.6, so I'm heading in the right direction. But look where I've come from. Look where I've come from. So we are continuing this journey as we improve our soils. So this is just in 1 year, and we are now progressing that again for this next year. It doesn't stop. We're keeping on that momentum. We mustn't lose that momentum as we move to where we need to be, to our 1-to-1 fungal to bacteria ratio. Okay, Carla, thank you. Right, guys, I'm going to whip you through very quickly some extracts and teas. So what we're going to do, we're going to be looking at forms of large and small scale of extracts, and the application and results we can get from that. Thank you, Carla. So look, there's loads of different ways of extracting microbes from our compost. I've seen people using concrete mixers, and then they can probably put seed in there as well to inoculate seed. Or you can use a nice bag there. We've got the bag, we've got compost, got some water in a little pail, and you can then start massaging the extract out. That's great. It works really well. Okay, Carla. And then you can start moving on to, if you're looking at high levels of or more quantity required, you can start moving on to this sort of system where you have a filter, you put it in a barrel, and then you put in your compost and you put water through it, and it flushes all that biology out of the compost through the filter and into a sprayer or an applicator of some description. Thank you, Carla. And then you can start moving to a even larger scale applications, and the picture there is of a screw auger type system that you put the compost in and water goes in the system as well. Or you can start doing this sort of system where you have an air blower, and inside that air blower, what we've done is we've made a round barrel with lots of air holes in it. You'll see it in a second, and we fill that up with compost. And then we fill that up with compost. We lift that container and we put it into the water, and we use on the floor there, you can see a plasterer's paddle, and then we mix it up within the water. I'm not going to let you watch the rest of this because we'll do that next time. So we can move on, please, Carla. Thank you. If it'll let you. And there we go. So we can start. We've now got 2 of these big IBCs, and we can start then also blending different types of compost. So I've got a compost that's fairly fresh and new, that's full of protozoa and nematodes, and then I've got a compost that's really heavy in fungi, but slightly lower in my protozoa and nematode levels. And I can then blend those 2 together in my bags and bubble them through to then start applying them to large scale tracts of land. Okay, Carla, thank you. And we've even got to a point where we're now applying this sort of extract directly into our seed as we're planting. So we don't inoculate the seed and put it in a bag and then store it away. We actually do it live. So we make it live, put it into the ground as we're planting. Thank you, Carla. Or you can then scope up and we can then start using very large scale extractors. This is a large machine that we've developed, or the box we've developed, and we're now putting extracts in, compost in that, and you can see the biology and all that great extract is coming over the top, and it's being mixed around, and that gets applied. If you go to the next slide, please, Carla. There we are. So it actually gets applied to the ground directly, and you can cover 100 hectares a day with a machine like that. Okay, Carla, and we move on. And then we start to make sure we're looking at how we're spreading that material. Very important, the spread pattern. I've used some of this spot-on paper, which gives me an idea of what coverage I'm making. But the little bit of paper on the bottom right is showing that would be 400 litres a hectare. That's what that looks like. That's before I bought the paper. Okay, Carla. And over to you, Brian.
Yeah, just quickly, I think it's this one slide. So going back to the compost application that I was starting to get into, but here we go. So again, 15 tons per acre per year. And then here you can see us spreading it. And as Nick mentioned yesterday, yes, it's very dry. It doesn't rain in California for many months. And we try to conserve some water, but we do irrigate regularly to make sure these microbes are thriving. And then 15 tons per acre, just for anyone that hasn't spread anything before, you don't see 15 tons per acre after it's been spread. It's hard to even recognize it's on the ground. You'll see the tire tracks from the tractor, and that's about it. You can find some pieces here and there. So it's not that much. You could do even more if you had it. We had done mostly broadcast spreading, like through the track, like kind of between rows and in the rows as well. Then we started banding it, so just getting it closer, concentrating that compost on the trees, and that helps a lot as well. High quality compost on wheat. Yeah, so as I mentioned earlier, we would set aside higher quality compost or create higher quality compost that we would save for new orchard plantings or weak orchards. And sometimes mixing this compost with worm compost again to get those same results. And we'll go more into this in the slide in the presentation later this month. The other thing I wanted to mention too, when I mentioned earlier, the marketing aspect of this is, especially when you're not selling the compost, this doesn't generate profit on a day-to-day business. You're in the business, especially in an orchard anyways, it's more obvious, but on any farm, you're considering results in the multi-year range. You can even be considering 5, 10, 30 years. You're trying to improve the soil. So any benefits you can get from it are great. Any financial benefits you can get up front are great. So we did create some teas. I applied these teas using a 500-gallon orchard sprayer. And what I would do regularly, especially in the spring when you're treating for fungal pathogens preventatively, such as here I've listed leaf curl on the peaches and the nectarines. You can get eutypa on apricots if they're pruned late. Brown rot on all the stone fruit and the mulberries. Powdery mildew on all the fruit. Fire blight, so bacterial, trying to inoculate are trying to basically get on and into the soil and onto the surface of the leaves and the branches. Again, tens of thousands of species of microorganisms, bacteria and fungi. So when a pathogen comes in on a storm front or just in the wind, it doesn't take over. It can't take over. It's outcompeted. It's consumed. It's just now part of this biome that you have that's very diverse. And then on top of all that is the fertility that you get from it. So most people consider compost as fertility. For us, or at least for me, that's the second thing I'm thinking of. I'm considering more the biology of what I'm putting out there rather than the fertility, because the biology is going to create the fertility that I need. I think that's all I have on that.
Brilliant. Next one then, Carla. Thank you. And I'll just reiterate what Brian said. So we apply our compost, if we're applying solid compost with a spreader and applicator, anywhere between 2 and 5 tons a hectare, depending on the biology. Again, put it under a microscope, understand what's there before you start spreading. And we only spread it 6 meters. And the reason we do that is because we're trying to be gentle with the compost. We have a nice curtain of compost coming out of the back of the spreader. Now that machine could quite easily throw compost 24 meters. That's going to obliterate anything. So we go at slower speeds, and the PTO is going really gently. So it's just giving us a nice gentle curtain. Because if you're doing a job and you've got all that lovely biology, why at the last step would you ruin it? So it takes a little bit longer and do it. And a bit like Brian, that's what 5 tons a hectare looks like on some soil. Right, Carla, I've brought it right back in. We are absolutely on 40 minutes for those 2 presentations. So whistle stop
Well
tour.
done, team.
But remember, guys, the 29th of April, Brian and I again will be talking about large-scale composting, and we'll have lots of time to discuss it. So please put it in your diaries. Let's see you all here for that event on the 29th.
Thank you so much, Nick. Thank you so much, Brian, Katerina. And while you guys catch up a little of breath, now we're gonna see a promo video from the school. And I hope you enjoy as much as we do.
Some of the farmers working with soil food web consultants around the world are seeing remarkable results.
Just within the first 3 years of working with these folks, they saved over $1 million just by cutting back on synthetic fertilizers, going from 200 units of nitrogen per acre down to 150 to even 50 units of nitrogen with the same, if not a better yield, which is really exciting.
If we look at the fertilizer savings across 1 hectare, we're looking at about $1,000 of savings. And then when we look at the increased revenue from the yield increases, we're looking at about $504. So the gross margin was increased by about $1,500. And then if you extrapolate this over 200 hectares, we're looking at about $300,000 over the course of any given year.
You can see the average again, 72%. And if you just take a few of those examples, you can take celery or you can take kale, you can see that we reach over 100% of yield increase.
Basically, the weight gain again was like 25% or more. The only complaint they got was the client wasn't used to obviously baling as often because the crop is growing so much better, so much healthier. They went from 280 to well over 400 sacks an acre. So that's 42% increase, which is really impressive. I mean, dollars and cents wise, that's a big jump. The PEs were really amazing. They were really aggressive with some of the PE projects. 136% increase. That's impressive.
Spring is in the air, and there's never been a better time to launch your career in soil regeneration than right now. This April, we're making the Springboard 2025 offer available for just $2,499. With the Springboard 2025 offer, you can register for the Soil Food Web Foundation courses for just $2,499, saving over $2,500 off the regular price of $5,000. You'll also get 2 free bonus courses, the Introduction to Permaculture and the Soil Sponge Workshop, worth $599. The total value of this bundle is $5,599, but you'll only pay $2,499, saving you $3,100. That's over 50% off. There are only 60 places available in the 2025 Springboard registration cycle, so please don't delay. Soil Food Web School offers a 100% money-back guarantee, so if you're not completely satisfied, you can get a full refund in the first 30 days, so long as you've watched 32 lectures or less of the foundation courses. Financing options are now available, so you can pay at your own pace with a firm. Join the soil revolution today.
Awesome. Ooh,
Some of the farmers working with Soil Food Web
bear with me. Okay, thank you. Sorry for this. So please take a quick shot on this QR code where you're going to find more information about this promo. It's the Launch Your Career in Soil Regeneration bundle. And you, as our guest, now have an additional 10% off. So please take this in your advantage and visit our website. Actually, this website with the QR code, and you're going to find the link in the chat as well. And if you have anything, any question, any doubt, you can email the school and someone will help you right away. So you can also email info@soilfoodweb.com for extra support. We hope to see you guys there if you're not our student yet. So please join us at some point. I just have to... Sorry, give me a second here. Oh, okay.
You okay, Carla?
Da-da-dum.
Technology.
Yeah. Come on, computer.
I did
The
so
slide
well.
deck just went
So
nuts.
well time.
Okay, I think I got it now.
Brilliant.
Okay, so panel, let's start our Q&A. It's always exciting and we have amazing questions. I was taking a sneak peek there, so prepare your brain and let's fire this up. So the first question is from Tanya. I would like to turn a flower bed into a vegetable garden. How do you suggest I amend it? Who wants to start?
I can start, but of course, that is just a starting point. I mean, as always, what I would suggest doing or what I would do is, first of all, checking what's going on in the present. So as we were saying before, we are like taking photos of the situation and what's going on in this moment. And so that what I would do at the beginning. So ideally check the soil, right? What's going on in terms of biology, where we are at. And then based on that, I would add compost accordingly, aiming for specific biological groups to be present and reaching a specific FB ratio, which is appropriate for the vegetables you want to grow. Then that is also a point. What do you want to grow and how diverse the vegetables garden needs to be and so on. But yes, first of all, check what's going on. And then I would say, but then Nick, Brian, and Carla, you may have more practical knowledge than what I have. I guess compost could be a good starting point because you're adding both the biology and the organic matter, right?
Yeah, and I think you've hit on a few things, Katrina, I would like to hit on. For me, whenever I'm doing anything like this and anyone asks me, I always think of that successional stage of soils and what plants do I want to grow, okay? And where do they fit into that successional stage? And where, as Katrina rightly said, and where's your soil at the moment? Because you might have too much fungi in there, you might have not enough, depending on the type of vegetables that you want to grow. So I think that's what I would do straight away. I would start looking at where your soil is currently and what your plan is for your brilliant veg patch that you're aiming to transform your flower bed into, and then start maybe finding someone who's got some great biocomplete compost, making sure that you check the biology before you buy any compost, I would suggest. So you and then introduce that as your soil results dictate to you.
I think you two hit the nail in the head, and it's a microscope is your best friend in this process, no matter what. Just like, and it's something that comes with practice as well. Don't stress out. You don't need to be an insane microscopist. Things will become easier as more time you spend in the microscope. And we have other amendments as well, right? We can use the protozoa infusion, we can use nematode extraction, the teas extract, and the compost. So all this variety is important, like Nick and Katarina said, understand where you are so you can define your next actions. But
Absolutely.
summarizing, the answer is yes, you can change any soil you want to adapt to the desired crop. It's just a matter of planning accordingly. Brian, anything to add?
I think that's all correct. I guess quick additions would be not all compost is equal. I think that was mentioned. So you're going to want to either have someone do analysis on this compost because it's going to make a difference. You can grow good food in not so great a compost because it's night and day difference still from the grocery store. So gardens versus grocery stores, night and day difference. But then there's another level. When you have a garden with microbiology, the fruit, the vegetables are even that much tastier. It's another shade, like completely different night and day shift. Because I've always gardened and the grocery store is always terrible. But then when you start gardening really well with biology or farming with biology, the bricks level of your food, the health of the crop is better, but then the bricks level, the nutrient content, I mean, it's crunchier, it's sweeter, it's just a whole other, it's a game changer. So yeah, check out the biology.
Yeah, and I'd like to echo what Brian's just said. You know, when I did my project, we did some lettuces and we planted some lettuce and put in some biology. And when we took some leaves from all of the lettuce plants that we had growing, we took it to be analyzed for the sap to see what nutrient levels are in the sap. And it was 10, 15, 20, 30% higher in those minerals that we're lacking currently in our diet. And that was through the compost and extracts that we were applying. And I know of another chap that's made a brilliant garden and brought in some compost and was very proud to see his compost turn up. Unfortunately, we put it under a microscope and it was just bacteria in it and lots of plastic, I would say, lots of plastic. So just be careful when you're buying or getting compost in that you know what you're buying. Really important.
That's a spot on. There is since you guys open up another realm, there is one, I'm going to present, representing the school in a workshop next month, beginning of next month, in 15 days, talking about exactly this connection between how improving the soil health, we can have better food, and this automatically reflects in human health. So science finally is connecting all the dots from different areas, and agriculture is no different. There is no way to not work if we are not interdisciplinary team. We need to understand the entire chain, not just the field to table process, but understand how everything is deeply interconnected. After all, we are all different species in this planet, so we are in symbiosis. And that amuses me that finally we reach that point. So it's another thing that I really encourage everyone to continue to follow up, not just understand how things happen in the soil, but how you can change that in your body as well. And that for me is one perfect spot to increase the sale price of your produce, and also as a consultant to get more clients based on this whole chain that you can expand.
Yeah, and all this knowledge, Carlo, is coming from the foundation course. You know, all this, it just starts to get you moving along that same route. You know, you engage into the foundation course, and it starts teaching you all of these things, which is so valuable when you're trying to turn a flower bed, however lovely they are, into a great veggie bed.
That's true. Yeah. So I think we can move to the next question. It's from Chase. Does anyone have experience using a Venturi injector to inject extract and tea into the line of a sprayer just before the spray nozzle? therefore bypassing the tank, pumps, right angles, etc. This would save the biology a ton of wear and tear, as well as being much easier to clean the system afterwards. Nick, Brian, do you guys have experience with this nozzle?
Do you want to go first, Brian?
I have experience with the nozzle, but not using it for teas and extra. I mean, I have done so I use the Venturi injector for putting certain types of soluble nutrients into drip irrigation. I have not done it with extracts. So and then I think mainly just because worry about clogging the drip line and having to clean that out. I just haven't got into it. I prefer to spray it. That's all I have.
Yeah, I think for me, depending on whether it's whatever method of applying biology through this method, which I think is a great idea, I think we just need to be really careful of maybe some antagonism between whatever you've got, your main carrier that's in your sprayer that's going through the lines. What is that you're putting on? Would you believe I was with a farmer not so long ago who was applying fungicides through a sprayer and then was trying to apply biology in, just as it comes out the jet. And I just told him, you are just wasting your time. So it all depends what you're trying to put it through. So just be very careful with that. Make sure there's no antagonism. It could be minerals. It could be fish hydrolysate. It could be all sorts of things. But just be really, really careful with that. And also the sprayer that you're using or anyone's using, just make sure that, I would never use anything with biology with a sprayer that's had some form of herbicide in it at any point, really. Even however clean you try and make that sprayer, even if it's the carrier of something and you're putting the biology with it, it just doesn't feel right to me. Again, going to all that effort of making your amazing extracts and teas and compost, and then it drops down at the last minute because the actual application doesn't work. I mean, one way of checking whether it does work when you do put it through these sorts of systems, which I do with mine, is actually again, it's going to go back to the microscope, guys. I'm sorry, but I test it when it's in the tank, and then I test it literally as it comes out of the nozzle, and we can then see the difference, because you will lose some, but it's just gauging what's physically going on. And just because you've got great levels in the tank doesn't mean you'll have great levels going out onto the field. And then when it doesn't work, you'll be kicking yourself that the system doesn't work. Well, it does, but you just need to make sure you know what you're putting on.
Yeah, and I think that's imperative to not forget to clean up as soon as you are done to avoid the biofilms, which can be a nightmare to clean as well. So it's just tiny adjustments that looks like daunting, but once you get to the flow in the field, it starts to make sense. And when you see all the difference in the results, worth all the changes you were making. Okay, so next one is from Alinani. How many different types of plants and wood material should be collected to make a biocomplete compost? For example, what plants or grains should I use to get the beneficial nematodes and protozoa in my compost?
I would say the more the merrier. We typically, in the coursework and CTP program for our first piles, we stick with one type of high nitrogen just to evaluate it. But the more you know about your materials, the more diversity, the better. And again, as Katerina mentioned, you're looking for an indigenous, local microorganisms. So you're considering materials that are available to you in your area. And in my opinion, yeah, the more diversity, the better. As long as you're not experimenting, because when you're experimenting at first and learning, you reduce the variables to learn about those materials, but as you learn about them, then you put in as much as you can. That's my opinion.
Yeah, diversity is the key, no matter which stage you are. So, and also you can use compost piles that you already have to inoculate new ones that you're building. So you're going to continue to build up in the diversity you have in the field. And you'll notice that diversity will varies from the source of the feedstock you have. You might be using horse manure for every single recipe, but if you change the source, you might have different biology depending on how the diet is managed from the animals, how they manage the animal health. So there is a lot of layers in this so-called diversity that we need to worry about. Anything else, Katarina, Nick?
No, I think we've covered all that one.
Yep.
Carla, that's great. Great question.
Another one from Charles. What needs to be done through proof of concept is educating the primary producer on the importance of soil microbes. You are doing that in spades, I believe you. How can farmers nurture, protect, and proliferate soil life in our soil system? Farmers are busy keeping their heads above water. We don't want to hurt our precious resource, but we are. Help us stop it. I would love to do all of what you're saying to do, but who will do all the other work we have to do in the farm?
Gosh, I mean, we've only got another 24 minutes to discuss this one, and I think this is a whole webinar question on its own, Carla, don't you think?
That's
Well, I'm going to pull it apart a little bit, I think. I mean, for me, Charles, great that you are thinking along these lines, and the biggest change for me is mindset, absolute mindset change, and I do believe that people are now starting to consider a change in their thinking on food production. And I can't wait to sit in Carla's webinar when she starts talking about all these microbes and what it's doing to our biome as well. It's going to be fascinating. And I think there are people that are really starting to look at that. I mean, look, I'm a farm manager as well. So Charles, I understand the pressures that we all have on our businesses, from labor through to cash, through to, you know, commodity markets and all those sorts of things. So I feel your pain. I have managed to do it, and we went cold turkey when we moved from our current model, which was conventional in Norfolk, where we are disturbing the soils, putting on fertilizers and chemicals, and we've made that change. We went cold turkey and did everything. I wouldn't do that I wouldn't do that now if I had my time again. But I would do, certainly, if your farm, however big it is, I would stick it into chunks and do slowly, so it's less of a big strain on resources, valuable resources you have on your farm, and it can be done fairly simply. You don't need to spend a huge sum of money. These round towers that we make of compost, I mean, you need a pallet, you need some round mesh material, and as long as you've got a spade or a fork and a brush and some water and the materials, you're there. And I make 6 or 7 or 8 of these every 3 or 4 months. I don't need to, because I can make windrows with my machine, but I still like to do this because I get better microbes, and then I can inoculate other things with it. So you'll be amazed how much you can inoculate with these small round towers. So I wouldn't give up the fight. It sounds like you're really keen to do something. Yeah, happy to, join the Soil Food Web and see how it's done. Again, join the foundation course, and you'll see how you as a farmer can change these things.
Yeah, I think you touched on a very good point. Everything needs to be well planned, and do not jump ahead of the horses. Just go slowly in your own pace, plan ahead, test in small plots, so you're gonna have a better idea of how much material you're gonna need. And it's just like, take your time. Don't feel that insane pressure to do all at once. And it's not a magical solution either. It's not one growing season, I'm done just maintenance after that. It's dynamic. Biology will change every single time you're going to take a sample in the microscope. So have that in mind. And like Nick said, the foundation courses are going to really help you understand all these bases that you need to focus on when planning that for your farm.
Do you know, guys, since, I mean, I did the foundation course probably 3 years ago now, maybe 4, and I still refer back to it, Carla. I still refer back to it. You know, there'd be lectures that I can remember through my notes, and I'll play the video again. And you just can't absorb everything in that foundation course in one go. It just keeps on giving as a piece of education material.
Elaine is just I mean, sorry about the term, but she's insane. The amount of information she can put in one lecture still amuses me after these 6 years. You're
Absolutely.
correct. It's insane.
I would add a
Okay.
thing. I think
Go ahead.
I agree with everything you said. I'm not a farmer, but one thing is that everything connected to agriculture and also as we said before today, the context is super important. So I would say, other than learning, as Nick and Carla said, because knowing the theory and the basic mechanisms is super important, also to start thinking about how you could apply those things in your context, but also like creating a network or getting in contact with other people that already like did that or tried that could be also very important. I think sometimes not only talking about something, it's enough. You need to see other people doing that close to you that have similar conditions. So I think if you can like reach out to people that you know that are quite close to you or have similar conditions and connect with them. And I think that could be very, very useful in a very practical way. And I think, yeah, that's the only thing I wanted to add.
And that's great, Katrina. And there's lots of people within the Soil Food Web that if you again join the foundation, you can reach out to such a great network of people who are going through similar sort of challenges, good ones and some excellent challenges, or some ones that are really difficult throughout this whole process. So I would, yeah, I can't speak highly enough of the foundation course because it's just giving so much, and the network of people is just amazing.
It is. Brian, anything else?
Yeah, that's good. And then, yeah, I think you mentioned I read the question several times. It's very interesting. I like it. Educating the primary producer for sure. We also need to educate the public. I mean, there's a demand for certain products, and that demand could change for organic, for regenerative, and that's a huge thing. And then also educating the voter. In the US, you have the Farm Bill. So once everyone's values change, your farmer becomes now the bank or like the car dealership or what other people value now. So I think it's a matter of just educating everybody. And the other aspect I was going to touch on was taking like as Nick said, yeah, taking the foundational courses changed everything I do. So even if I'm not making compost, what I originally did was I was more excited just to have learned how important soil microbes are and how they function. So I wasn't even interested in making compost at first. I just didn't want to destroy the microbes I had, and you can nurture the soil and get microbes growing. It just takes longer if you're not going to use compost. So I initially got into raising beef cattle and rotationally grazing them, and during that whole time just paying attention to what was happening with the soil, what was happening with my cows, never making any compost or spreading any amendments. So I think just the foundational courses that it'll start to change your thinking and other things will open up. So again, yeah, couldn't recommend the foundational courses anymore.
So let's go to another question from Stenbergen. Sorry if I'm not saying your name correctly. How long does the compost recipe take to decompose and becoming ready to be used in soil? I would start as if Elaine would be here. It depends, but normally it can take about 3 to 6 months in thermophilic stage. Who wants to compliment?
Katerina, go on. I bet you've got a lot to add for this one with all your experience.
I think that's right. It depends. And actually this is something that after trying, as also Nick showed us, no matter how big you're composting, trying in smaller sizes, it's also very important because that is a factor you may think it's a major aspect you want to control, right? When we plan for making compost and practice recipes, right? We think about also like time, how much time consuming it can be for you, right? So if you want it to be faster in terms of like decomposing, that is something you want to keep in mind when practicing and trying recipes, right? And so based on that, you're going to plan or think about starting material that can decompose faster, yet giving you not only a successful pile, but also a biocomplete pile, right? Or full of biology. So yes, it depends and it takes practice and it depends on your goals and what are your objectives and main pillars, let's say, right? That's it, I guess. I don't have much more to say.
Another thing is that you said it's the level of biology you want to achieve in a compost depending on the crop you have in the field. So that's where the depends also takes in place. In time of the year, location of the planet you are. In tropical zone, things will go much faster. In temperate zones, it will take a bit slower, but it's about how to adapt the recipe as well to kind of speed up the process. But you said beautifully, it's about, and like Brian said from the previous question, is the whole process we work here is to speed up the recovery. So it's just like giving nature an extra compensation for all the damage we did so far. Anything else on this?
Yeah, I mean, for me, making large scale windrows, or seasonality is a big one for me as well. That is incredible. So in the spring, it makes very quickly, as long as I get my moisture right. As long as I get my moisture right, absolutely. We have taken a pile once where I got my moisture wrong, and it dried out after the second turn, going through those time and temperature heat stages we talk about, and it dried out. So I thought, I know this is a great opportunity for me to try something. And I let it dry nearly right out. And then I gradually introduced water to it. It took over 100 liters a meter of water to get it rehydrated, which was insane, absolutely insane. I'm glad I did it, but I wouldn't do it again. And the compost started going through the heating cycles again when it got to that 40-50% moisture. Then we were back up and running and going again. And it still made really quite quickly. In the autumn, that's why I have some extra nitrogen in forms of clover. So my normal materials I would use, I put those on and I kind of sprinkle a bit more clover on the top, a bit more legumes on the top to make sure I get through those 3 cycles. Because I've been to a situation before where I've gone to the second cycle and it's just died away. So it's all about the material, and also remember, guys, about the seasonality. When is the sap rising? Is it starting to senesce? And how that affects then your compost.
Spot on. So the next one is from Sarah. I'm going to farm a land for a farmer who is too old to do it himself. He's currently farming in a non-organic way. I'm currently trying to learn what is the best way to farm the land. Recommendations.
Again, everything just depends. What was he growing? How was he growing it? How non-organic was it? All those things. And then, yeah, starting with, again, I couldn't recommend the foundational courses more highly to just have a biological understanding in a very short, just very quick time to learn that. And that connected so many pieces for me. I know I had farmed for about 6 years before taking those courses, and I and Nick did it even longer. And I think the first thing I think when I'm taking this course is like, oh, why didn't I take this earlier? Because it would have helped so much. And but yeah, going to the question, it really depends on what was growing. What do you want to grow? What are you farming? Do you have cows? Do you have oats, flowers? It depends on many things. And then again, we mentioned networking, network with people in the area that have grown similar in that climate and maybe perhaps similar crops, and they can help out as well.
And I would agree with everything that Brian said, and Sarah, as a consultant in this situation, and I see many farmers that are either giving land up or want to change the way they farm. And it's really great if the farmer still wants to be involved in some way, just see what he wants to do, get him involved. It's about understanding his context of the farm and what he's done, as Brian said, in the past, and what he might like to do in the future. So he's on board with what you're trying to do. And if you're trying to farm in this kind of way, then it'd be great for him to be on the journey with you. So it kind of works in tandem. My boss, as soon as I started the foundation course, lo and behold, he jumped on the foundation course as well, because he's not a farmer. He is not a farmer. He's a million miles away from being a farmer, but he wanted to understand it, right? And that was great for me because I had a buddy who I could talk to everything about. We understood what was going on, and I felt that he had a buy-in to this journey that we're on. And the journeys doesn't take sort of one application or two applications of extract. 50, 100 years of damage, we're not going to solve it with one application of extracts. I can assure you. So this is a 2, 3, 4 year journey, depending on how hard you go. So I would try and take him on the journey as well. Yeah, that's what I would do. That would be my advice.
Hook up the microscope on a big screen while you're doing the assessments and let him have fun with you.
Absolutely.
So, okay. Let me just check if we don't have more questions just because the system is. Bear with me just a second.
I didn't add any more questions. That's the end. There
Oh, okay.
More though. You guys went through them
Thank
quickly.
you, Sam.
Well, Sammy, so Carla, I think it'd be really great to run round and just kind of see what with a 5-6 minutes we have left, just some thoughts from this brilliant webinar, and it's great to have Katrina and Brian with us. Just their kind of final wrap-up thoughts of today's webinar. Who would like to go first? Brian, I'm going to throw you under the bus first.
Yeah, I deserve it, I guess.
Earlier, I'm going to get you back.
Final wrap-up thoughts. I mean, the first thought, I'm glad to connect with people that are interested in soil and producing good food. And I think, like everything, it's a journey. So finding a place to get started, finding somewhere, to get started on that journey, connect with other people, getting started. And today was to me, the webinar was a very high level overview of what's covered in hours and hours and hours and hours and hours and hours and hours of lectures, and then hands-on experience. So yeah, I think just everyone to have that in mind. That's what I would take away from this, is that this was a very high level overview. And if it spoke to you, if it got you excited, the foundational course is a great place to start. But even if not, go talk to a farmer, go to a farmer's market, go connect with somebody that's growing food, or start a garden, do something. That's my thought, which wasn't my thought up, but good. Thank you.
No, brilliant. That's absolutely great. And now Katrina's had time to think of a great response as well to that question.
I mean, Brian said it all. I would agree. I mean, I was thinking about my perception as being part of the community already, and also how I would have perceived this webinar being outside or just interested. And I think from one side, it could like feel a little bit overwhelming because as Brian said, it was a lot, right? Even quite enough detail of information, like from microscopy and then how we do build piles and make teas and extracts, and also the practical aspects. So a little bit overwhelming, but at the same time, I think we needed a webinar such as this one that was like really like, I don't know, connecting the dots, right? Connecting these aspects and giving a broader overview so that whoever wants or is interested and knows it can start with foundation courses, but knows what's the process looks like, and also the diversity of approaches you can go for. So yeah, I mean, that was very exciting for me, and I hope that was exciting for everyone else joining today. So thank you. Thank you, Carla and Nick, for great like hosting. So yeah.
If I mention
Thank you
one
very much.
more thing,
Oh,
I guess the
yes.
one thing I didn't mention, my background is not farming at all. So I started farming about 10 years ago. I came from engineering and business consulting and had no experience, did not grow up on a farm, never farmed, just loved food, loved cooking. And that's where I got connected, farmers markets, questioning farmers about what they were growing, volunteering for vegetables, just to get my hands literally dirty and started out. So, I mean, it's definitely a great place to go into that's growing. And you don't have to be it actually has been extremely helpful to me not to have studied agriculture or have worked around farmers, because my mind was completely plastic and able to bend to what needs to be done. Thanks.
Yeah, that's a great one, because it's great seeing people who have not a connection from a young age coming into the industry, because your mind is just way open to any opportunities that that you can see, and you're not kind of nervous to take those, Brian. So I absolutely agree. And for me, I think this has just been a great taste of what you might, if you were to join the Soul Food Web School, be getting yourself into. And it is honestly, for me, it was life changing. It's given me a business, it's given me a community to share and guide and help anyone who's on this amazing journey we're finding ourselves on now, to try and heal this damaged land and produce food that is not just quantity but more about quality, to help our health as we move through these next few years. And I suppose for one little take home for me, and I'm going to mention it for probably the 10th time now, anyone who's got a microscope and they want to be testing their soils, just always use your microscope. It is just an amazing piece of equipment. You don't have to be super amazing at it. You pick up things really quickly. And again, there's a community out there who can guide and show and help if you don't understand the difference between a testate amoebae and a naked amoebae or whatever it might be. And what does a pneumycete difference to a fungal hyphae? So it's all those things that I hope you will all embrace, and not be nervous about coming forward and trying to progress your career or knowledge into this amazing school and everything that Dr. Elaine has set up for us.
Yeah, and if I have something to complement, I would say that's never too late to start.
No.
Just respect your boundaries, your limits, but don't be afraid. Just
look how old I am.
White hair can be deceiving, Nick.
I dye this, you know. I'm really sort of brown. This is dyed. All the fashion, you know, all the fashion.
That's true. And I'm still amused on how universities, I know that this was a conversation in the chat, how universities are still lacking to talk about biology. We learn about food web in general since K-12 grade. We all learn about how soil produces grass, that rats come and the snake comes and all that stuff. And soil food web is essentially that knowledge. Actually, is that knowledge, is that basic ecological knowledge, where we just black that out from the higher level academic lectures and practicing the field. And that what attracted me to Elaine the most. One of the main things is that she promotes this bridge between the knowledge and to practical field. How we can take all this beautiful knowledge that stay in black boxes in academia and transform that in actions that anyone can do. And when we say anyone, it's anyone. If you guys can take a sneak peek in the background from our students, we have people from the most diverse background, learning how to work with the microscope, learning how to make compost. And like Brian said earlier, have a lot of people helping you make compost. They're gonna make just more fun and easier on you as well. But the message here is change mindset is crucial. Be open. Regenerative ag is a set of tools, and soil food web is one of the main one, but it's a non-stop learning curve. So don't be afraid, just jump in. We'll be here to help you whenever we can. And I just wanna remember you guys that next week we're gonna be together for the open house. So please bring all your experience, all your energy. We wanna hear from you. We wanna listen, you guys, and you guys give us so much energy back. You have no idea how this is incredible and how this makes us continue to translate and communicate Dr. Elaine's words. Who asked us to give you guys her warmest hug and appreciation for you continue to do the job. And we want to just make sure that you guys are following us in our new TikTok channel and Instagram, Facebook, and YouTube. Mark us when you post anything related to Soil Food Web. We love to see that. And we have a beautiful team helping us scan and check all of this, which I also want to extend the appreciation to our amazing staff that makes all those webinars possible. So thank you, Soil Food Web crew. Thank you, our amazing, beautiful students. And we hope to see you guys next week.
Take care, everyone. Thank you for joining us.
Thank you.
Stay safe.